Neurons and neuroglia. Some of this is going to be reviewed for you guys when we did histology. But there's a lot of stuff that's not. We're going to just expand with it. And then we'll talk a little bit about a review on action potential stuff and maybe just a few other things, and I'll let you guys go. Okay, neurons and neuroglia. There's two main types of cells in the nervous system. The neuron, all right, we've done that. There's three parts to them. The dendrites, the cell body, or the soma, and the axon, okay? But we're going to see that there could be different types of neurons, and we'll get into that in a moment. There's about 1 trillion of these in our body. And then we have the neuroglia. And the neuroglia is more of supportive tissue for the neuron. The neuron is the one that's going to carry the action potential, the jolt of electricity from one end to the other end going someplace. All right, when you plug in that wire for the lamp, you plug it in, and the jolt of electricity goes down the metal wire to go to the lamp and turn it on. That's the neuron that does it, the metal wire itself. But then we have neuroglia that's going to support, to make sure that that jolt of electricity gets from one point to the other point. A good example of this is the insulation. The insulation around that metal wire. That insulation is going to be neuroglia, because it's going to make sure that the electricity is not going to escape out of that wire. It's going to be contained within that wire. So the neuroglia is not going to conduct electricity. It's not going to have anything to do with generating or carrying an action potential. It's just going to support the neuron to allow the neuron to do its job with the action potential. We have about 50 neuroglia for every one neuron. So we have tons of these compared to the neurons. These are what can become cancerous, because it's not the neuron that would get cancers as you've seen in histology, but other things. And in a case like this, the neuroglia can. Now we have 6 different types of neuroglia, and we're going to get into them. We have astrocytes. I'm sorry it's very dim, but you'll see them on the next few slides. Astrocytes, microglia, Schwann cells, and epidermal cells. Those 4 that I just named are located in the peripheral in the peripheral nervous system. Then we have oligodendrocytes and satellite cells. These are located in the I'll show you which ones are in the peripheral. They weren't those. I'll show you them in a second. But we have 6 of them. So we have oligodendrocytes and satellite cells. Okay, so all these are here. I'll correct myself when you see the next few slides. You'll see. All right. Cancer. Like I said, mature neurons, extremely low cell turnover. They don't go through mitosis. It may take about 70 or 80 years from one neuron to actually turn into 2 neurons. God bless you. Okay. So these don't become cancers. However, there are neurons that are immature. And these would become cancers if they do, but it's only found in children, because in children, the neurons are still dividing. They're still immature and becoming mature. So those are still dividing. It's a rare cancer called neuroblastoma. If you see the word blast in there, it's usually a cancer of children. Retinoblastoma is cancer of the eye. Nephroblastoma is cancer of the kidney. Neuroblastoma is cancer of the neuron. And if it has blast in it, it's usually a cancer of children. Okay, not that adults can't get kidney cancer, but that specific one, nephroblastoma. Okay, but neuroglia, that's the majority of brain cancers that you see in adults. Okay. So that's what usually happens. And you could also think that not just neuroglia, but also like the pituitary gland. I mean, that's a part of the brain, but it's a gland. It's epithelial tissue. It's not the neuron that's actually becoming cancerous. Okay? So this is just an FYI, just to give you an example, but you have an astrocytoma, which is an astrocyte of a neuroglia cell that becomes cancers. an epidioma, which is an epididymal cell that becomes cancer. These are all cancer cells that are not neurons, they're neuroglia. So just to show you on this, so if someone says they have brain cancer, and God forbid, but brain cancer, you got to say, well, what do you mean by brain cancer? I mean, that's probably not something you would want to ask the person because they have other things to worry about than to know the type of it. But if you're going into the medical realm, you would have more of a curiosity, an interest of which one this person has, if it's one of your patients. Because I doubt very, very much it's not the neuron that becomes cancerous, it's the neuroglia cell that did. So there's a few of them over here, and there's a few over here. Okay? Again, these two charts are really just an FYI, just to emphasize that it's neuroglia cells that become cancers, not the neuron. So let's talk about the neuron itself. Again, there's three parts to a neuron, which you know about: the dendrite, the axon, and the cell body. And it's going to actually generate and propagate an action potential down the axon. Okay? There's going to be myelin around here. In our analogy that I use, with a wire that you're going to plug in for the lamp over here, the myelin is going to be the insulation around the wire itself. That's neuroglia cells that are doing that. Well, that plastic that goes around there is the myelin, okay? And that forms, as I said before, the white matter, because it has like a whitish color to it. And that's going to make sure that the action potential goes down the axon and it doesn't escape the axon. Because if I shred some of that plastic around the wire and remove some of it, then when you plug it in, it's going to find another way, the action potential or the electrical impulse is going to find another way to escape and not go to the lamp. It finds a shorter route to escape out of there. Okay? And then when you turn on the light, it's either going to be very dim or it doesn't go on at all because electricity escaped before it actually got to the lamp. And what is that disease where we remove the myelin? That's MS or multiple sclerosis. So they're not going to be able to move certain things. They're not going to be able to swallow. They're not going to be able to breathe if it gets so severe to the diaphragm they can't contract it and inhale. So that's what they usually die of, okay? Or they'll get dizzy or blurry vision. A bunch of different things. It's because that action potential can't get there. So a dendrite, a cell body, and an axon. I don't want to spend too much time on that because we've already exhausted everything about that. But I do want to say the cell body is also known as a soma, a neurosoma, or a perikaryon. If you want to master A and P, you got to learn how to say one structure about 12 different ways. It's all there, okay? So, all right. And an axon hillock, that's a new word for you. An axon hillock is where the action potential is initiated in the cell body, okay? Just before it gets into the axon. We call it the axon hillock. So let me show you what that is, right? You know what the run-of-the-mill kind of neuron is. Here's the cell body, here's your dendrites going all out there, and this is your axon. So the area of the cell body that's just before the axon, that's called the axon hillock. And then you're also going to have the first part of the axon that doesn't have myelin on it, but it's part of the axon. That's called the initial segment. You'll see the next few slides that I have it all defined there. When we put the axon hillock and the initial segment together, we call that the trigger zone. Okay? Just terms. This is just another picture showing the same thing, and then we'll talk later about the neuromuscular junction. Okay? Now here's something new. We've been drawing the neuron in a certain way, but let me just explain to you that's not the only way neurons are. There's 4 different types of neurons. The one I drew for you is what we call a multipolar neuron, because there's a lot of poles to it. You have many dendrites, but only one axon. So when you look at the cell body, there's a lot. It looks like a star. There has a lot of things coming out of it. Multipolar. Okay. Then we have bipolar. Bipolar is that there's one dendrite and there's one axon, and they kind of look like this. The cell body's here. The impulse is going that way and it's going out that way. So there's the dendrite and there's the axon. We see them mostly in the eyes and a little bit of the ear. Okay. Then we have unipolar, and unipolar is what we see in the dorsal root ganglion. When we get into the spinal cord, we're going to see a situation that's like this. Here's the cell body, and there's one that comes out like this. So there's only one that comes out. Dendrites here, the axon's over here. So it's only one that comes out. We call that unipolar. And you'll see that in where the nerve root is. When we get into where the spinal roots are. When we get into the spinal cord, you'll see that. Then we have something called anaxionic. Now, I don't know if this really exists or not. It just doesn't make sense to me. Some books have it, some books don't. But I'd rather just explain it to you so that when you get into other advanced courses, at least you've heard something about it. But I'm not saying it doesn't exist. I just don't understand it. It just doesn't make sense how that would be there. And it just is emphasized by some books not even doing this. All right? It just has many dendrites but no axon. I just don't understand when this would be used. It says it helps with visual processes, but I can't even see in the visual aid where this would be located. And I've seen different pictures, and so I don't know if it really exists or not. Okay. So these are just showing you different types. This is the one we drew before, so that's in multipolar. Here's the bipolar, and here is the unipolar. The anaxionic neuron, ana means without, axon is without axon, so it's just dendrites. I just don't understand where. So it goes here. Now where does it go? It doesn't make sense to me, okay? And I can't find the actual place in the eye that this exists, okay? So that's why some books it shows it, some books it don't, okay? All right, we have three classes of neurons, right? We have sensory neurons. Things go into the central nervous system. And we have about 10 million of those. We have interneurons, which we have abundance of. And they're all located in the central nervous system. That's the only place they're going to be located in there. We see a lot of them in our brain. All right, those are what's going to connect the sensory neurons with the motor neurons. So the last one is the motor neurons. We only have about 500,000 of those. That sounds like a lot, but when you compare it to the other things, that's just a little fraction. Okay? All right, so let's talk about the different types of neuroglia. Okay? We have 1 trillion neurons and 50 trillion neuroglia. They're there to support and protect the neurons. They're not going to actually produce an action potential or propagate an action potential, but they're surely going to make sure that the neuron is doing their job. So here's where it is. So here's the CNS. These are the 4 neuroglia that are found in the central nervous system. You have epidermal cell, microglia, oligodendrocytes, and astrocytes. And in the peripheral nervous system, you've got the Schwann cells and satellite cells. Okay? Now let's talk about each one of these. I have the next 3 slides. I'll have 2 of them on there explaining it. First one, the epidermal cells. The epidermal cells, these are the ones that are going to line the cavities in our brain. And yes, our brains do have hollow cavities. They're called ventricles. Some people's are a little bit more hollow than others, but we do have hollow cavities in our brain. The lining of these cavities is going to be make cerebral spinal fluid. When you get into, I think it's the Frankenstein PowerPoint presentation, it'll talk about cerebral spinal fluid. But cerebral spinal fluid is going to bathe the brain and spinal cord, and it's made by these epidermal cells. Okay? And that's what's that. Then we have microglia. It's a type of macrophage. Macrophages used to be certain white blood cells called monocytes. And what happens here is monocytes are like the police that are patrolling the streets and making sure everything is good out there. And, crime is down. And then the commissioner says, you know what? We have too much crime in our local mall over here, so I don't want you to be patrolling the streets anymore. I don't want you to go into the bloodstream. I want you now to be stationed in an organ. And I want you to be stationed in the brain. So you're going to be what we call a macrophage. You're going to be now patrolling inside the brain and stay in there for the rest of your life, and you're going to be looking for viruses and bacteria. So you're a macrophage. But in the brain, this macrophage has a special name. It's called microglia. We have macrophages in the liver. They're called Kupffer cells. We have macrophages in the lungs. They're called dust cells. We have macrophages in the kidneys, and they're called mesangial cells. So macrophages are all over. I won't ask you about those, but macrophages are found in different organs. Okay? And they're going to destroy pathogens like bacteria and viruses. Now, we also have oligodendrocytes. This is going to create myelin in the central nervous system. And we have one cell over here that's going to wrap itself around an axon and also be able to wrap itself around another axon and be able to wrap itself on another axon. So you have one oligodendrocyte that can actually create myelin on 10 or 20 different axons all at the same time. The oligodendrocyte can do that. We also have our Schwann cell, but they can only wrap around one axon at a time. And these are found in the peripheral nervous system. It kind of looks like, same way up here, if let's say the axon itself is think of Christmas wrapping paper. It's that cardboard dowel that's in the center, and then the wrapping paper wraps and wraps and wraps and wraps and wraps. That's what the Schwann cell and the oligodendrocytes are going to do. They're producing myelin that's going to wrap around that axon. Okay? It's just one is in the central nervous system, the other one is in the peripheral nervous system. Then we have something called an astrocyte. An astrocyte is the most abundant neuroglial cell and connects the neuron to the capillaries. This is going to build something that we call the blood-brain barrier. This is going to block certain chemicals, a lot of chemicals going into the brain. It's going to block bacteria and viruses going into the brain. Now this is a double-edged sword. It's doing a wonderful thing. It's blocking the bacteria from getting into the brain and viruses. But it's also going to limit what medications can get into the brain also. There's a big problem with a lot of the psychological medications for bipolar, schizophrenia, all those kinds of medications that need to get into the brain. Parkinson's disease, that kind of stuff. Because the chemicals, the medications need to be able to break through that blood-brain barrier to get into the brain. Fortunately, it takes many weeks to get into the brain. You got to get a big dosage, or you got to put a huge dosage, and unfortunately, a lot of times the amount that gets into the brain is only maybe about 5 or 10%. So the other 90% hit receptors throughout the rest of the body that might be the ones that need to target inside the brain. And because of that, you get a lot of side effects. So that's why pharmaceutical companies, they're relying on getting across the blood-brain barrier to get into the brain. Okay? Now there's certain areas of the blood-brain barrier that are not there. In other words, there's got to be a little opening there. Places that need to have access from the brain going into the bloodstream. Places where the brain makes certain hormones. So an area where the pituitary gland is, there's going to be a break at the blood-brain barrier so that the hormone leaves the pituitary gland and enters the bloodstream. It won't have to go through the blood-brain barrier. Does that make sense? It's a break. These breaks are what's keen for you to understand, because this is where HIV finds those little breaks and gets inside the brain. But it's a double-edged sword. We need those breaks, otherwise we can't function. We can't get certain hormones coming out of the brain. But it's also an area where bacteria and viruses know where those breaks are. Okay? Questions about that? Okay? And then our last one is the satellite cell. This is found, as we talk about the blood-brain barrier, this is found in the central nervous system. The satellite cell basically does the same thing, but it occurs in the peripheral nervous system. It creates the, I wouldn't call it the blood-brain barrier, but a barrier nonetheless from the environment going into the peripheral nerves. Okay, and that's what happens over there. So this is a big picture shown here. Here's your oligodendrocyte that's going to actually put myelin on a few different axons. We're going to have the astrocyte over here where the capillaries here, so it's producing this blood-brain barrier. So it's kind of, this picture is kind of putting a lot of things together that we just talked about. Okay? So those are our 6 different neuroglia. You should know the differences between them, almost like make your own chart if that's going to be helpful for you. Which ones are in the peripheral nervous system, which are the central nervous system, what's the functions of each. Okay? Questions on that? Okay. So let's talk a little bit more about myelin. Okay? Myelin, as I explained to you, and this picture's not really doing its justice up here, it kind of got watered down from the screen. But if you look at the computer slide, you'll see that this area is blue. This is a myelin, and I'll show you more pictures of it. But the myelin sheath is over here. Okay? Like I said, here's the axon. If you look at a cross-section of it, think of it like a Christmas wrapping paper. There's that cardboard dowel, the axon, and then you're wrapping paper all around here, more and more and more, to make it really thick. That's basically what you got here. Okay? Now, a few things. It's insulation. It creates that white matter, is what we call it. The gray matter is unmyelinated areas. Now, there are some axons that are unmyelinated, but all the dendrites and all the cell bodies are not myelinated, so that's also going to be gray matter. Okay? This insulation allows the electricity or action potential to go from one place of the body to the other place extremely fast. We call it a saltatory conduction. The myelin makes for fast conduction of that action potential. It's not continuous. In other words, we got one myelin Schwann cell over here, so to say. We have one over here, and we got another one over here. But there's a gap in between. I'll show you that in a moment. The areas of the exposed axons in between the myelin is called nodes or rhombier. Okay? And like I said, they have this saltatory conduction, makes it go very fast when it's myelinated. The completion of this myelination doesn't really occur until late in adolescence. But I can show you, and that's why we definitely don't want to have kids drinking alcohol and doing drugs at an adolescent age, because the nervous system is still developing. Does that make sense? It's not completed. But let me show you what I mean by a 1-year-old, so you can understand the difference presentation-wise when myelin is not on something. I think you could relate to this. Think of a 1-year-old, okay? And if they're obedient enough and you could ask them to do something, and do this next time you see it. Use your child as an experiment, guinea pig, all right? But it's no harm, it's no harm. If you put uncooked rice on your table, you know, the hard rice, that kind of thing, and you put them on your table, and you ask the child to pick it up. Yeah, come on, pick it up. Come on, pick up the rice. Come on, pick up the rice. Come on. And you show him, and then he'll want to do that. She'll want to do that, right? But how is he going to pick it up? How does he get his hand and pick up that rice? You're all doing it. I know you are. They're not picking up like this. Not a 1-year-old. They pick it up like this, right? Why? Because the myelination has not been formed at the fingertips. There's no sensation. They get no gratification by picking up that. It feels like there's like Elmer's glue that's dried up there. They feel pressure, but they don't feel tingly by picking up things. The palm of their hand, they do feel that. And that's what they like. They can feel it. They can feel, they can only feel pressure on their fingertips. But on their palm of their hand, oh, that's really enjoyable, because that's a new feeling that they have. It's like intense, and they like to pick that up. So that's why they pick it up. Does that make sense? Okay? So that's a good example of what I mean by how myelination makes a difference that you could actually visualize. So this is a close-up. This is, here you got myelin here. Let's talk about Schwann cells. Like this is a Schwann cell, this is a Schwann cell, but it's not continuous. There's a gap there. There's an exposed area of axon. Does that make sense? Okay? And what happens here is that this area of exposed axon is called a node of Ranvier. And the best way I could explain why it goes so fast, it's a saltatory conduction. It goes like this, like this. I don't like the arrows pointing this way, because they really go this way, inside. The axon potential doesn't go out, it stays in. The best way I could explain this is, let's say you want to, if this is water in here, and from this point here to this point here is the length of an Olympic-sized swimming pool. And we are going to cover the swimming pool, the whole thing, except at the very end and the very beginning. Okay? You're going to jump in here, and you want to swim and swim and swim, and you want to get up for air. So you're going to swim faster and faster and faster to get to where the opening is, and you pop up for air. And then you do it again. So what's happening is, for myelination, you're able to actually swim faster, or the action potential goes faster when there's myelination there. Does that make sense? You can visualize something like that. It's the best analogy I can come up with. Okay? But there's areas here. This is where the action potential occurs. What goes on here is that there's no sodium coming in over here. It's only at the areas where there's exposed areas. But the conduction goes very fast. It jumps from one node of Ranvier to another. Okay? And it doesn't go backwards because it's in a refractory period before. Okay? Again, you can see nodes of Ranviers over here. Right? And just to give you another term, just to throw it in there, that's a node of Ranvier. The area of the axon that's inside the myelin, we call that an internode. Okay? So you have internode, node of Ranvier. Internode, node of Ranvier. So it's another word. You'll see in our next few slides, it's called internodes. Oh, there it is. All right. So the internode is the area of the axon that's covered by the myelin. Okay? The initial segment, we'll go back to this picture here. The initial segment is the first part of the axon that does not have the myelin on it. And when you put the initial segment and the axon hillock together, that's called the trigger zone. Okay? So it's the area that's going to initiate or manufacture the first action potential. Okay? White matter, as I explained to you, has myelination. Gray matter, no myelination. All right? And as I said before, the gray matter surrounds the white matter. In the spinal cord, it's the opposite. The white matter surrounds the gray matter. Diseases of myelin. Does anyone know who that is? I'm kind of embarrassed putting these people up here because you guys don't even know these people. She's kind of like the first Mouseketeer. It's kind of like Miley Cyrus, but back in the 1950s and 60s. Annette Fucciello, no? Okay, well she had a certain disease. Anyone know who this is? Richard Pryor, good, okay. Comedian back in the 80s or so. Terry Garr from Young Frankenstein. We'll move up to something here. Who's this?
The talk show guy.
Yeah, you're on the right track. But who doesn't do the talk show anymore? Montel Williams? Okay. This one my parents wouldn't know, but you guys would. Jack, Jack Osbourne. So I know Ozzy Osbourne, and we know what drugs can do to you now, okay? But the drugs have nothing to do with what he's got. So this is Ozzy Osbourne's son, Jack. What did all these have in common? Yeah, they have multiple sclerosis, MS, okay? And again, what it is, it's an autoimmune disease. Why the person makes these autoimmune antibodies, we don't know. but they make antibodies that attack the myelin, where it attacks the oligodendrocytes. So therefore, it only attacks myelin in the central nervous system, not the peripheral nervous system. It doesn't attack Schwann cells, it attacks the oligodendrocytes, which is found in the central nervous system. There's a possibility, we're not too sure, but it could be triggered by a virus. It doesn't cause a virus, but what I'm saying here is that if you have the gene for multiple sclerosis, and I'm just giving you numbers, you're supposed to get MS at, let's say, 30 years old. If you have a certain virus, it might kick it on 5 years before than what you should get it. You won't know when you're supposed to get it, but there's some talk about a virus actually speeding up the process of when you're going to get the disease, if you carry the gene for it. Does that make sense? Okay, we're still wondering about that, but there is some research that suggests that, but it's not a proven thing. Okay, so myelin gets replaced with this hardened scar tissue. The scar tissue is what we call sclerosed tissue. Okay, so you have multiple areas of this sclerosis, and it's going to disrupt, because the sclerosis, the scar tissue, kind of gets hard and presses down on the axon and disrupts the action potential from going from one place to another. And that's what's going to happen. They get double vision, I don't know why it's popping up, double vision, tremors, numbness, you get all these kinds of things. More females than males get this, and the onset is between 20 and 40 years old. Usually when you get diagnosed with it, you're looking at about 25 years until death. Okay, so that's, some people have beaten that, sure, they have, but the rule of thumb is once you diagnose, it's about 25 years. Okay, and they usually die when the diaphragm is not working anymore. Okay? So that's what happens, that's what it looks like normally, but gradually it starts breaking down. And that's what multiple sclerosis is. Okay? So it's demyelination in the central nervous system. Tay-Sachs disease, we talked about this when we did cytology, but Tay-Sachs is a genetic disease that a lysosomal enzyme called hexosaminidase, and what this does, it's an enzyme that usually will break down glycolipids from accumulating. Okay, glycolipids, glucose and lipids together, they kind of break them down. But if you're missing this enzyme, then the glycolipids will increase, and we see a lot of it increasing in the brain. And when there's a lot of this, it's going to start pressing down on the axons and the neurons, and it's going to disrupt, and it's going to interfere with the conduction of the action potential in there. And that's what's going to happen. So we usually see it in acidic Jews. We see that a lot over there. It leads to blindness, loss of coordination, dementia, and it's usually death by 5 years old. Okay? So that's where that comes in. Remember we talked about Tay-Sachs? Speed or velocity of a nerve signal, this is pretty common sense, right? Large diameter fibers, you would expect to go faster. So if you have large fibers, nerve fibers, it's going to go faster. If you have myelination on there, it's going to go faster. Okay? So you could bet that large myelinated nerve fibers are going to be very fast, whereas small, or let's say thinner fibers and ones that are unmyelinated, they'll go slower. Okay? And that's what I'm trying to show up here. You don't have to worry about the millimeters per second, but it just gives you some idea of that. Okay? Functions. Slow signals are used for supplying the stomach and dilate the pupil. You don't want the pupil to go really fast. You don't want the stomach to squeeze the food forward very fast. You want it to have some time for it to get absorbed. Right? Fast signals will be skeletal muscles, so you can get the heck out of there. Right? It's also going to transport signals for vision and balance. You know, if someone pushes you, want to be able to get on that balance quite rapidly. Okay? So we'll talk more about that when we get into other parts of the nervous system.